Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. In question 1, we have a photomicrograph showing a few cells at different stages of cell cycle. 1 asks you to identify the stages of mitosis in B and C. B shows chromosomes that have condensed but are still arranged almost circularly in the cell. This is the prophase, where mitosis has just started. In C, the chromosomes align into a row. They are now at the equator of the spindle. So, this is the metaphase. 2. Describe the behavior of the chromosomes in the stage of mitosis shown in cell A. Cell A shows 2 groups of chromosomes. They are moving towards the opposite poles. It is a cell in the anaphase. During anaphase, sister chromatids of the chromosomes are pulled by the spindle fibers and separated. They start to move to the opposite poles. You can also call them daughter chromosomes, as each of the chromatids is a chromosome on their own now. The movement is due to the contraction of spindle fibers that are attached to the centromeres of the chromosomes. Centromeres divide at the start of anaphase, so now the sister chromatids have 1 centromere each. The centromeres are leading the movement as it is the part that attaches to the spindle fiber. 3 wants you to describe the role of DNA ligase in interphase. DNA ligase is an enzyme that is active during DNA replication. This means they are active during the S phase. It joins the short fragments called the Okazaki fragments together on the lagging strand. Okazaki fragments are defined as the small sections of DNA which form during discontinuous synthesis of the lagging strand during DNA replication. The synthesis of DNA cannot occur continuously at the lagging strand, as DNA polymerase can only make DNA in the 5' to 3' direction. Ligase catalyzes the formation of phosphodiester bonds between the fragments. Note that even though DNA polymerase also catalyzes the formation of the phosphodiester bonds, it joins the free activated nucleotides together, not fragments. Before the action of ligase, there are gaps or nicks between the fragments. Its action completes the sugar-phosphate backbone as the nicks are sealed. B1. State the location of telomeres on a chromosome. Telomeres are found at both ends of a chromosome. 2. Suggest why a long lifespan could result from a higher telomerase activity. To answer this question, you must know the function of telomerase. Telomerase is the enzyme responsible for the maintenance of the length of telomeres. It catalyzes the addition of short single-stranded DNA repeats to the end of the chromosomes. If their activities are higher than normal in a cell, telomeres will be maintained for a longer length of time. The shortening of telomeres limits the number of cell cycles a cell can go through. If they are maintained, the cell can undergo more DNA replications and cell cycles. During DNA replication, DNA polymerase cannot attach to the end of the DNA, so it cannot complete the synthesis of the new strand. This results in the DNA getting shorter after each replication. Telomeres, which do not code for a protein, help to prevent the loss of essential genes. This is why, when telomeres are critically short, a cell cannot undergo the cell cycle. Note that telomeres can be described as genetic material. Stating that they prevent the loss of genetic material is incorrect. If our cells can divide more times, we will have a greater ability to replace the damaged, worn out or old cells in our bodies. This provides us with a longer lifespan. Besides, when telomeres are replaced, a cell does not age and will not go through apoptosis. Apoptosis is the process of programmed self-death, which is used to eliminate those unwanted cells in our body. 2a shows how the number of TB cases changed in the USA from 1980 to 2000. 1. Describe the trends shown in the figure. When you are asked to describe a trend, always start with a general statement to give an overall idea of the pattern shown in the graph. In this case, there's an overall decrease in the number of reported cases throughout the years. Then, quote the data from both axes to support this statement. Separate the graph into a few distinct parts and describe each part using the data. In the beginning, there is a drop in the number of cases. Then, it is followed by a period of slight changes. The number increases afterwards before decreasing again. Remember to use comparative values to support these points. In my answer, you can see that I always quote the starting and ending values in the descriptions. You can get 1 extra mark sometimes by doing so. Remember that in your data description, you must use precise values. Do not use terms such as around and approximately to answer this question. 2. Explain how an increase in the number of people infected with HIV could have affected the number of TB cases. HIV is an infection which leads to a weakened immune system. When a person's immune system is not as effective, there will be an increased risk of latent TB become active. This is because the bacteria cannot be suppressed efficiently by the immune system. You can get 1 mark by giving the details of how HIV weakens a person's immune system. The target cell of HIV is T helper cells. The infection would destroy T helper cells, causing a decrease in their numbers. If there are fewer T helper cells to release cytokines, there will be a poorer humoral response, as it will be more difficult to activate the B lymphocytes to produce antibodies. There is a mark for AVP. For example, if there are more people infected with HIV in a population, there will be more people with TB, hence increasing the risk of the spreading of the disease. B wants you to state 2 ways that the impact of antibiotic resistance can be reduced. The command word state means you should give precise and straight-to-the-point answers. Elaboration and detailed explanations are not required. When the question has mentioned the number of answers you should provide, you don't have to give any extra points as they will not be marked. First, antibiotics should only be prescribed when it is necessary. This is to prevent the overuse of antibiotics, which will increase the exposure of bacteria to these chemicals. Doctors should make sure that a correct and effective antibiotic is prescribed. This is to ensure that all pathogenic bacteria are eliminated. New antibiotics should be developed to prevent the same antibiotic from being used too frequently. We should also consider the use of other antibacterials. Some non-antibiotic drugs, such as anti-inflammatory and local anesthetics, have shown antibacterial activity in some studies. They may be used as an alternative to treat bacterial infections. Besides, we can use other measures to prevent an infection and break the transmission cycle. Such measures include vaccines and good hygiene in the hospitals. This will help to reduce our reliance on antibiotics as a treatment. Cases and patterns of antibiotic resistance should be reported. Updated data can help scientists and health authorities to understand the cause and determine effective ways to overcome the issue. Antibiotics in agricultural farms and animal use for food should be regulated and controlled. This can reduce the chance of bacteria in the human body encountering antibiotics in the food. Education for healthcare professionals and the public must be done. This allows everyone to understand the role they can play in reducing the impact of antibiotic resistance. C1 suggests the effect that damage to cartilage may have on the bronchi. Cartilage provides mechanical support for the bronchi. When it is damaged, bronchi lose this structural support and may collapse. C2 wants you to pick the cell that is present or absent in each of the structures. The alveolus is one cell thick. The only cell it has is the squamous epithelial cell. The flattened shape of this cell provides a short diffusion distance, hence a greater efficiency for the gas exchange. The bronchus and trachea are very similar. You can think of the bronchus as a smaller version of the trachea. They have all of these structures except the squamous epithelial cell, as their epithelium is made up of ciliated epithelial cells and goblet cells. Figure 3.1 shows a blood smear. To identify the types of white blood cells in a photomicrograph, you can use the shape of their nuclei and their sizes. X has a multilobe nucleus. You can also see granules in the cytoplasm. These are the features of a neutrophil. There are 2 acceptable answers for Y, monocyte and lymphocyte. Even though we can't see the horseshoe-shaped nucleus, judging from its large size, this is a monocyte. Do not name it a macrophage, as this is a blood smear. Monocytes only develop into macrophages in the tissue. Due to the circular-shaped nucleus, identifying it as a lymphocyte is also credited. However, lymphocytes usually have the same size as red blood cells, so cell Y is not likely a lymphocyte. A2 wants you to calculate the magnification used to produce the image. The formula is Magnification equals to image length over the actual length. Use your ruler to measure line Z in the photomicrograph and express it in millimeter. Then, convert it to micrometer by multiplying 1000. Divide it by 8 micrometer and you will get the magnification. B1. Describe how carbonic anhydrase is involved in the release of oxygen from red blood cells at respiring tissues. At the respiring tissue, carbon dioxide is released due to respiration. They will diffuse into the red blood cells. The enzyme will then catalyze the reaction between carbon dioxide and water in the cell. Carbonic acid is formed. Then, it dissociates to form a proton and hydrogen carbonate ion. Proton will bind to the Hem group of the hemoglobin to form hemoglobinic acid. It displaces the oxygen which was attached to the Hem group. Hemoglobin has a higher affinity towards protons, so it has a higher tendency to bind with it and release oxygen. The association of protons with the hemoglobin causes a conformational change in hemoglobin. This shape change reducing its affinity for oxygen molecules. It is known as the allosteric effect. In 2, we have to sketch a graph when a competitive inhibitor is present. The rate of reaction is lower when the substrate concentration is low. This is because competitive inhibitors bind to the active site, reducing the chance of ES complexes formation. However, competitive inhibition can be overcome by an increase in substrate concentration. So, the graph will approach or achieve the same Vmax as the original graph at a high substrate concentration. Question 4 is about gluten, which contains 2 proteins. 4a wants you to match the description in the table to the level of protein structure. Whether a protein is compact depends on how the polypeptide folds to give the final 3D shape. It also relies on how different polypeptides in the protein interact. Since gliadin is a single polypeptide, the tertiary structure is the one that contributes to this feature. The second statement gives information about the amino acids in the protein. So, this is the primary structure, the sequence of amino acids in a polypeptide. The last statement mentioned beta-pleated sheets. It is related to secondary structure. 2 states that the genes code for gliadin do not contain introns. You are asked to explain how these affect the production of mRNA from the primary transcript. To answer this question, you must know what introns are. They are the non-coding sequence in DNA. After transcription, introns are removed from the primary transcript. The coding parts called exons will be joined together, forming the mature mRNA. If a gene does not contain introns, RNA splicing is not required. There is nothing to be removed from the primary transcript. The joining back of exons is also not necessary. So, the primary transcript would have the same sequence as the mRNA. Without introns, alternative RNA splicing cannot be done. This is a process where exons from the same genes are joined in different combinations, forming different mRNAs. So, only other post-transcriptional processing, such as 5' guanine capping and the addition of poly A tail, would take place. B talks about the celiac disease. 1. Suggest and explain how gliadin peptides are transported into intestinal cell. The question mentioned that gliadin peptides are often as large as 33 amino acids in length and are taken up by cells in large quantities. To take up a molecule of such a large size and in a large quantity, cells need to carry out the bulk transport called endocytosis. You can also say that it is by pinocytosis. Any detail of the process will give you a second mark. There will be the invagination or infolding of the cell surface membrane. Parts of the membrane will fuse to form endocytotic or pinocytotic vesicles. This is an active process which uses ATP. B2. Describe the sequence of events that results in the immune system producing anti-gliadin antibodies. An immune response occurs because of a non-self antigen. In this case, gliadin acts as an antigen. There might be an antigen presentation carried out by antigen-presenting cells, such as macrophages. The antigen is recognized by specific B and T lymphocytes with complementary receptors. These lymphocytes are activated. The process is called clonal selection. Then, they will undergo clonal expansion. The lymphocytes start to divide by mitosis. The activated T helper cells secrete cytokine. Cytokine is a cell signaling molecule which stimulates the B lymphocyte response. You can also call this the humoral response. Some of the B lymphocytes differentiate to form plasma cells and start to produce antibodies. This question is about the production of antibodies, so you do not have to include the actions of other cells such as killer T cells and the formation of memory cells. 5a wants you to label the phloem tissue and xylem tissue. The three oval-shaped structures are vascular bundles. Xylem and phloem are located inside. You can separate the tissue in a vascular bundle of a dicot stem into 4 layers. The outermost layer, which is nearest to the epidermis, is the sclerenchyma. It is a supportive structure. You can also call it the phloem fiber cap, but remember, it is not phloem. The layer after this is the phloem. They look more feathered as they have thinner cell walls. Then, we have cambium. These cells can divide and differentiate into cells in the xylem or phloem tissues. The layer nearest to the center is the xylem. They have the largest cells with thick cell walls. You can also see large lumens of the xylem vessel in this layer. B1 describes how sucrose is transported in phloem sieve tubes from photosynthesizing leaves to other parts of the plant. The question is not asking about the whole loading process, so you don't have to talk about how sucrose moves from the mesophyll into the companion cell. Sucrose is transported in phloem sieve tubes by mass flow. Mass flow is the movement of fluids down a pressure gradient. Sucrose diffuses into the phloem sieve tube from the companion cell through plasmodesmata. There are many plasmodesmata between the cells, allowing an efficient movement. Sucrose is a solute. It is dissolved in water and lowers the water potential inside the sieve tube element. This causes water to move into the phloem sieve tube by osmosis down the water potential gradient. The volume increases in the sieve tube element, which leads to an increase in the hydrostatic pressure. At the sink, sucrose is unloaded, and water follows by osmosis. This is because the removal of sucrose from the sieve tube element decreases the water potential inside. The hydrostatic pressure will decrease as well. A hydrostatic pressure gradient is set up between the source and the sink, and transport occurs down the hydrostatic pressure gradient. B2 says that the cyanide ions inhibit the activity of an enzyme involved in respiration. Suggests why the treatment of photosynthesizing leaves with cyanide ions results in less sucrose being transported into phloem sieve tubes. Respiration is a process where energy is released from respiratory substrate such as glucose to produce ATP. If an enzyme in the reaction is inhibited, there will be a lower rate of reaction. Less or no ATP is produced. So, less ATP is available for sucrose synthesis in mesophyll cells. Active transport of protons out of companion cells for the loading of sucrose will also decrease. Without the proton gradient, less sucrose is co-transported into the companion cells. A lower sucrose concentration in companion cells leads to a decreased diffusion of sucrose into phloem sieve tubes, as the concentration gradient is less steep. C is about the Benedict's test. The question wants you to suggest which of the two solutions would change color in the shortest time. When photosynthesis occurs in the mesophyll cells, glucose is synthesized. It will then be converted into sucrose before being transported in the phloem sieve tube. So, A would change color in the shortest time because the sugar present in the cytoplasm of mesophyll cells is mainly glucose, a reducing sugar. In solution B, the sugar present is mainly sucrose, which is a non-reducing sugar. Benedict's solution does not react with non-reducing sugar, so the time taken for color change in solution B would be slower. 6a shows an oligosaccharide called nistose. You have to state 3 differences between the structure of nistose and glycogen, other than the number of monomers in the molecules. Nistose is a linear chain with no branch. Glycogen is branched. Being a linear chain, nistose has only one type of glycosidic bond, which is 1,2 glycosidic bonds. Glycogen has 2 types of glycosidic bonds, 1,4 and 1,6. 1,6 bonds occurs at the branches. There are 2 types of monomers in nistose. Glycogen has only one type of monomer, which is alpha-glucose. Nistose contains only one glucose residue, unlike glycogen, which is made up entirely of glucose. Lastly, You can compare the ring structures of their monomers. B. Describe the roles of the rough endoplasmic reticulum and the Golgi body in synthesizing glycoproteins. Rough ER has ribosomes on its surface. Translation occurs at the ribosomes and polypeptide is produced. The polypeptide will fold in RER's lumen into its specific shape. The protein is then packaged into transport vesicles. The vesicles will transport the protein to the Golgi body. In the Golgi body, modification of the protein occurs. A process called glycosylation takes place for the formation of glycoprotein. Carbohydrate is attached to the protein in this process. If a non-protein group is needed in the proteins, the addition of it may occur here as well. These processes are known as post-translational modification. The final product is then packaged into secretory vesicles and transported to the cell surface membrane. Both organelles can check for any misfolded or incorrectly synthesized proteins. The faulty proteins will be degraded. C. State one role of glycoproteins in the cell surface membrane. Glycoproteins can act as receptors for signaling molecules such as hormones. This is an important part of cell signaling. They can also act as cell surface antigens. This is for cell recognition, where our immune system can determine whether a cell is self or non-self. Glycoproteins are also involved in cell-cell interactions. For example, they help adjacent cells to adhere together. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.